Antibiotic resistance is a big problem in the field of medical science. So to understand that a little bit better, first we need to understand what are antibiotics and how do they work. There are two overall general classes of antibiotics. They're bacteriostatic and bactericidal. Bacteriostatic will slow down the bacteria. It allows them to be slowed and not able to replicate. Bacteriostatics, because of this, are slow acting. You're not going to directly kill the bacteria, you're just preventing them from reproducing, from making further generations, and so it's a bit slower than if it just outright kills them. This is a good thing, though, because it allows the immune system to launch a full innate and an adaptive immune response. So that way, if exposed again in the future, hopefully your body will be able to fight it instead of needing antibiotics. The only problem is that it does have a higher risk of allowing for antibiotic resistance, because since it is slower, you're going to take the antibiotic over time for a bit longer at lower doses. So there is the risk of bacterial antibiotic resistance, but that's why you should always finish your medicine, and we'll talk about that later. Bactericidals, side means to kill, and they target the essential components of the bacterial cell. They have the potential to completely eradicate the bacteria, resulting in a rapid clearing of the infection. This can lead to a very quick recovery. It has a lower risk of antibiotic resistance because you're not giving the bacteria a chance to survive and just not reproduce. The bactericidal is outright killing the bacteria, and that's why it does it rapidly, and there's a limit to the risk of infection. One thing to note is that it is a common misconception that static is not as good as cidal, that you want a bactericidal to clear the infection rapidly rather than the bacteriostatic. But recent studies have shown that that's not the case. Recent studies have shown that in life-threatening infections in patients, that bacteriostatics actually work better in the long run. They believe it's because it's allowing the immune system to do what it does best. It's giving the immune system a chance to kick in and an upper hand. And once the immune system kicks in, nothing works better than our immune system. There are different methods of action for these antibiotics. The first way that the antibiotics work is by targeting the DNA. And so they can inhibit the topoisomerase and the gyrase. If you remember, the topoisomerase is the enzyme that is responsible for unwinding the supercoiled DNA, while the gyrase is what unwinds the double helix, the double-stranded. So the topoisomerase allows us to have access to the DNA. The gyrase separates it into the two strands so polymerase can come in and do its job. There are also some antibiotics that not only inhibit the topoisomerase and gyrase, but they also are effective in eliminating plasmids. I have listed at the end there some of the antibiotics that we've seen in lab, as well as if they are a bactericidal or a bacteriostatic. This is going to help with your analysis, so make sure to pay attention to that part. Another way that antibiotics work is by affecting the permeability. Essentially, it pokes holes in the cell wall, which causes a leaky cell, and that leaky cell will leak its internals out and leading to death. If I poke a whole bunch of holes in you, and I'm not talking like poking small holes, but like inch-size holes all around you, eventually it's going to cause you problems and you're not going to survive. So the way that it does this is that in some cases it will block PBP proteins, which are penicillin binding proteins, and that's an enzyme that helps in cell wall stability. So it helps in building the peptidoglycan structure. The way that I explain this is it's kind of like, think of that peptidoglycan. I said it was like a bunch of little strings, just crisscrossing and overlapping. Well, if nothing's holding them together, then, you could easily do a light breath and scatter the threads all over the place. So the penicillin binding proteins are kind of like little dots of hot glue in a pattern, and that allows it to hold together and be stable. Or if you kind of think maybe of it like a chain link fence, if the wires were just put across just straight, it wouldn't be very effective. But by crisscrossing them at those crisscross points, that's where it's holding it stable and strong. And so the PBPs would be those crisscross points. Another way that it can, antibiotics can affect the permeability is just by straight blocking peptidoglycan synthesis. Peptidoglycan is a protein, and so if it blocks that processing specifically of peptidoglycan, then it will cause it to be leaky, it will have holes, and the cell can die. Bacitracin is an interesting antibiotic because it can be used either as a bacteriostatic in low concentrations or a bactericidal in high concentrations. The others are pretty honed in on what it is that they can be used for. They don't have quite as big of a range. Folic acid. Folic acid are things referred to as sulfa drugs. We had our example in class of a sulfamethoprene, and it's static. Folic acid is very important for production of red blood cells, as well as for cell growth, DNA repair, and DNA synthesis. Because of this, if you block a bacteria's ability to produce folic acid, the bacteria really can't make itself. It really can't reproduce itself again, and that's why it's static. It is already fully functioning, but you're putting in antibiotic that prevents it from making these things that will allow it to replicate itself and copy. So one bacteria stays one bacteria till it gets old and dies. It can't make any other bacteria. The last one is protein synthesis, and this gets utilized a lot. If you remember from our protein synthesis unit, the ribosome is responsible for translation, and the ribosome has two parts. The smaller 30S ribosome, which has the E site, P site, and A site, which are the elongation site, the peptide site where your peptide bonds form, and the A site or activation site. It also consists of the 50S ribosome, which was the larger portion. It's bonded to the 30S ribosome, and the 50S ribosome is kind of what catches the amino acids as they come into those EPA sites. So some of the antibiotics that we used inhibit the synthesis by binding and blocking one or more of those sites, the E, the P, and the A. Whereas others prevent bacteria by binding to the 50S ribosome. And by doing that, it prevents peptide bonding. The amino acids aren't able to join to the site. They can't form the peptide bond, and so they come in as individual amino acids and they leave as individual amino acids. So no protein is made. Hopefully this helps with this stuff. As far as which method of action is best really depends on the type of bacteria, whether it's gram-positive or gram-negative. Some of these will work best against gram-positive, some work best against gram-negative. For example, the permeability antibiotics. If you remember, our gram-positive has that large peptidoglycan outer layer. And so the permeability antibiotics work better on your gram-positive infections because they have direct access to that peptidoglycan. On the gram-negative, it's not nearly as easy because you have that outer lipid bilayer that is going to be semi-permeable, so it kind of protects the peptidoglycan layer. It's a little harder to get at with those drugs. They also will use a combination therapy, and so combination therapy is where they might use 2 different types of antibiotic to kind of do a double hit at it. So maybe they're going to use amoxicillin and novobicin. So the amoxicillin is going to poke holes in the peptidoglycan layer, and the novobicin is going to eliminate plasmids and inhibit telomerase and gyrase. So hopefully that makes sense. And like I said, I did list our antibiotics that we used in class here. So that way you can use this to help with your lab analysis and how it is that certain antibiotics were more or less effective on your bacteria. And hopefully this will help you come to some conclusions.